The first step in passaging cells is to make sure that you wash your hands so that you can try to be as sterile as possible. We want to limit any chances of contamination, and this is an easy first step to accomplish that. Double check to make sure that you have all your materials provided to you by your instructor. The next step is to make sure the hood you are working in is as sterile as possible. We start by using 70% ethanol and wiping down all the surfaces that might be covered with unwanted bacteria. This minimizes the risk of contamination to your cells that you are trying to culture. You want to grab your flask full of cells given to you by your instructor and the media that either they provide you or that you made yourself. You can start by loosening the lids to make your life a little bit easier. Push the pipette through the back of the wrapper to open it and push it into the pipette gun. We move the cap like that because it is a common sterile technique to reduce contamination. We next remove the old media out of the flask, discard the media and the pipette. We open our Falcon tube, which has trypsin in it. We get 2 ml from the Falcon tube and add it to our now empty flask. We use trypsin because it breaks down the proteins which enable the cells to adhere to the flask. Following common sterile technique, we make sure to place our lid appropriately and make sure we try to not contaminate our cells. We wait 10 seconds after adding the trypsin and take it over to the microscope to assess if the cells are detaching from the flask. After seeing that they have not detached, we open up another pipette and take 1 ml of the trypsin out of the flask while leaving 1 ml of trypsin in. We then go into our media with a new pipette and take out 2.5 ml of media and add that to the flask. We do this to deactivate the trypsin. We then take it to the microscope and examine if the cells have started to rise up off of the flask. Before splitting into new flasks, it's important to make sure you get a cell count so you know how much media you have to add into your flask. For each flask, we want 3.5 ml total in the flask. We have determined that we need to split our cells into 2 flasks, so we are going to add 3.5 ml of media to make a total of 7 ml in our flask. Using sterile technique, we change pipette tips each time they touch the cells. Now that we are splitting, We take out 3.5 ml from the flask and add that to a brand new flask. We continue to follow common sterile technique with our lids and our pipettes. We make sure we don't pass over any open containers with our arms. Try to imagine bacteria rain falling from your arms, getting into your culture, which might contaminate it. We repeat the first step and take out the rest of the 3.5 ml from our original flask and add that to a brand new flask. Make sure to tighten your lids so that there is no chance of spilling and contaminants. Tightening the lids too hard won't kill the cells because these lids provide gas exchange for the cells. However, if the lids are screwed on too tight, it might make it difficult for you to take them off the next time you try and open it. Now that our original flask has nothing in it, make sure to discard it in the proper biohazard waste container. An important step before incubation is to make sure that you have your flask labeled so you don't use someone else's or vice versa. Label them with the proper date, cell type, and passage number. Before you set your cells in the incubator, make sure that your liquid is covering the entire surface of the flask so you can get proper growth of your cells, demonstrated here. Take your cells to the incubator and place them in. Make sure you close the doors and seal everything shut behind you. To clean up, put your media back into the fridge, wipe all your surfaces down, and place everything back in its proper location before exiting the lab.